Basics of Medical Biochemistry. First edition, 2022. Chapter 7. Bioenergetics. Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tau, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 7.9 Complexes of electron transport chain, etc. The mitochondrial electron transport proteins are clustered into complexes known as complex I, II, III, IV and ADP synthase complex as complex V. The electron transport assembly catalyzes sequential oxidation-reduction reactions. Some of these reactions are thermodynamically competent to support ADP production via ADP synthase. 1 Complex I, it is composed of the enzyme NADH plus H plus dehydrogenase, NADH plus H plus coenzyme Q reductase. It is a huge complex, 850,000 kD, and composed of more than 30 subunits. It is a flavoprotein containing FMN and Fas as coenzymes. The prosthetic group FMN is absolutely required for its activity. It oxidizes NADH plus H plus and transfers electrons through FMN and Fas to coenzyme Q, which is reduced to coenzyme QH2. During electron transfer, protons are pumped from the mitochondrial matrix to the space between the inner and outer mitochondrial membranes called intermembranous space. The stoichiometry is 4H plus transported per 2 electrons. Later on, when protons pass through complex V, 1 ADP is produced. Figure 7.13 Structure and function of complex I. Figure 7.14 The sequence of electrons transfer in respiratory complexes. Source: Textbook of Biochemistry by Mary K. Campbell and Sean O'Farrell 8th ed. 2 Complex II, it is succinate coenzyme Q oxidoreductase, succinate dehydrogenase. It is the only membrane-bound enzyme in the TCA cycle. It contains Fad and Fas. This complex is composed of 4 subunits, 2 of which are iron-sulfur proteins and the other 2 subunits together bind Fad through a covalent link to a histidine residue. These 2 subunits are called flavoprotein 2 or FP2. It lacks the proton pump ability. It reduces coenzyme Q by the following reactions. Succinate plus Fad fumarate plus FADH2. FADH2 plus coenzyme Q Fad plus coenzyme QH2. Ubiquinone, coenzyme Q, and cytochrome C are mobile carriers. They ferry electrons from one complex to the next. Electron flow through the mitochondrial electron transport assembly is carried out through several enzyme complexes. Electrons enter the transport chain primarily from cytosolic NADH plus H plus to mitochondrial NADH plus H plus, but can also be supplied by succinate to mitochondrial FADH2, or by the glycerol phosphate shuttle via mitochondrial FADH2. Reduced coenzyme QH2 diffuses in the lipid phase of the membrane and donates its electrons to complex III. For complex II, the standard free energy change of the overall reaction is too small to drive the transport of protons across the inner mitochondrial membrane. This accounts for the 1.5 ADP generated per FADH2 compared with the 2.5 ADP generated per NADH plus H plus. Figure 7.15 Glycerol phosphate shuttle, DHAP, dihydroxyacetone phosphate, PDH, phosphate dehydrogenase. 3 Complex III, cytochrome BC1 complex or ubiquinone cytochrome C oxidoreductase. It is composed of CytB and CytC1. Complex III is a crystal structure. There are 2 B-type cytochromes and 1 C-type cytochrome. It catalyzes the transfer of electrons from coenzyme QH2 to CytC1. Cytochrome B contains the same iron protoporphyrin as hemoglobin and myoglobin. The Bl heme is present near the P face. The Bl heme transfers its electron to the Bh heme. Subscripts L and H refer to high and low potential, which is found near the N face. The cytochrome C contains heme C through covalent attachment by cysteine residues. The RISC protein, which is an iron-sulfur Pfau protein, is mobile in the crystal structure of complex 3. This mobility is required for the electron transfer function of this protein. During the transfer of electrons, protons are pumped across the inner mitochondrial membrane to intermembranous space. Later on, when protons pass through complex V, 1 ADP is produced. Figure 7.16 Structure of complex 3. For complex 4, cytochrome C oxidase or ferrocytochrome C oxygen oxidoreductase. It is a multi-unit structure containing cytochrome A 3 with heme A and 3 copper ions. 2 of the copper ions form a center called Qu, while the third copper ion is called Cub. It transfers electrons from reduced cytochrome C to O2 through cytochrome A and A3. Iron and copper are involved in oxidation/reduction. Heme A and Cu act together to transfer electrons to oxygen. The copper undergoes a transition from Cu+ to Cu2+ during the transfer of electrons through the complex to molecular oxygen. Oxygen is the final electron acceptor, with water being the final product of oxygen reduction. During electrons transfer, protons are pumped across the inner mitochondrial membrane to intermembranous space, as it also functions as a proton pump. Later on, when protons pass through complex V, 1 ADP is produced. Figure 7.17 Structure of complex 4, Source, microbiologynote.com. Figure 7.18 Flow of electrons in the electron transport chain and oxidative phosphorylation, Source, jackweston.com. 5 Complex V, ADP synthase complex. It is a multi-subunit enzyme scattered over the surface of the inner mitochondrial membrane. ADP synthase is composed of 3 fragments, F0, which is localized in the membrane, F1, ADP synthesizing head, which is protruded from the inside of the inner membrane into the matrix, and oligomycin sensitivity conferring protein, OSCP, which connects F0 to F1. F1, it has 3 alpha, 3 beta, gamma, delta and epsilon chains. It contains the phosphorylation mechanism. Peripheral catalytic sites are present on beta subunits. F0, it is composed of A, B and C units. The C units are 10 to 12 in number and present in the form of a ring. It forms the proton channel. It is embedded in the membrane and acts as a channel for the transport of H+. Synthesis of ADP occurs on the surface of F1. The binding change mechanism states that 3 beta subunits change conformations during catalysis, with only 1 beta subunit acting as a catalytic site. Beta subunits of complex V occur in 3 forms. IO form, open form, it has a low affinity for substrates ADP and pi. 2L form, loose form, it can bind substrates ADP and pi but catalytically it is inactive. 3T form, tight form, it binds substrates ADP and pi tightly and catalyzes ADP synthesis. When protons pass through the disk of C subunits of F0 unit, it causes rotation of the gamma subunit. The gamma subunit is in the form of an axle, it rotates when protons flow occurs. The beta subunits, which are fixed to the membrane, do not rotate. ADP and pi are taken up sequentially by the beta subunits, which undergo conformational changes and form ADP. Figure 7.19 Structure of complex V, ADP synthase complex. 7.10 Oxidative phosphorylation. The process of biological oxidation is essential for the vitality of all living tissues. During this oxidation, a part of the released energy is utilized in the synthesis of high-energy phosphate compounds such as ADP, so-called oxidative phosphorylation. The free energy produced as a consequence of the transfer of 2 electrons from NADH plus H plus or succinate to molecular oxygen is 57 and 36 kcal/mol, respectively. Oxidative phosphorylation traps this energy as the high-energy phosphate of ADP. The following 2 conditions are required for oxidative phosphorylation to proceed. 1. The inner mitochondrial membrane must be physically intact. 2. A high concentration of protons must be developed on the outside of the inner membrane. The chemiosmotic potential or proton motive force, PMF, is developed by the energy of the proton gradient. This potential is the sum of the concentration difference of protons across the membrane and the difference in electrical charge across the membrane. A gradient of 6 protons is generated by the transfer of 2 electrons from NADH plus H+. The oxidation of 1 mole of NADH plus H+ leads to the availability of a PMF with a free energy of about 31.2 kcal, 6 times 5.2 kcal. The energy of the gradient is used to drive ADP synthesis when the protons are transported back into the mitochondria. Figure 7.20 ADP synthesis by ADP synthase complex at F1 results from repetitive conformational changes as gamma rotates. Source: Molecular Cell Biology, 6th edition, 2008 W. H. Freeman and Company. 7.11 Chemiosmosis theory. The chemiosmotic hypothesis was proposed by the British biochemist, Peter Mitchell. This theory explains the mechanism of oxidative phosphorylation in mitochondria, and photophosphorylation in chloroplasts. He was awarded the Nobel Prize in 1978 for this valuable work. This hypothesis stated that a proton motive force is responsible for driving the synthesis of ADP. The chemiosmotic hypothesis is based on osmosis, the diffusion of water across a membrane, that's why it is called chemiosmosis. This theory attempts to explain the conservation of free energy in this process based on an osmotic potential caused by a proton concentration differential, or proton gradient, across a proton-impermeable membrane. It allows protons to pass through the membrane and uses the free energy difference to phosphorylate adenosine diphosphate, ADP, to make ADP. Mechanism: 1. Complex I, 3 and 4 act as proton pumps. They transfer protons from the mitochondrial matrix to the space between the inner and outer mitochondrial membranes called intermembranous space. As a result, the intermembranous space becomes more acidic and more positive, and an electrochemical gradient is developed called the proton motive force, PMF. 2. The proton motive force, PMF, drives the synthesis of ADP by the ADP synthase complex. ADP synthesis is an endergonic process needing inputs of energy. 3. The protons, H+, flow back across the inner membrane through ADP synthase resulting in the trapping of free energy as ADP. 4. One hydrogen ion enters the ADP synthase complex from the intermembranous space and a second hydrogen ion leaves it in the matrix space. The upper part of the ADP synthase complex rotates when a new hydrogen ion enters. 5. Complex I, 3 and 4 are involved in the transfer of electrons from NADH plus H plus to CIT A plus A3. These complexes are responsible for the pump of 4, 4 and 2 protons respectively. 6. Complex II, 3 and 4 are involved in the transfer of electrons from FADH2 to CIT A plus A3. These complexes are responsible for the pump of 0, 4 and 2 protons respectively. Complex II does not pump any proton. 7. The transfer of 4 protons is equivalent to the synthesis of 1 ADP. The total number of ADP produced per NADH plus H plus is 2.5 ADP as it transfers 10 protons, whereas the total number of ADP produced per FADH2 is 1.5 ADP as it transfers only 6 protons. 8. The ADP phosphorylation and electron transfer are coupled by the electrochemical potential of protons. This mechanism of ADP synthesis is called oxidative phosphorylation. Figure 7.21 Mechanism of chemiosmotic theory. 7.12 Regulation of oxidative phosphorylation. The rate of ADP synthesis is mainly regulated by the energy needs of the cell. When energy demands are increased, during vigorous muscle activity and protein synthesis, cytosolic ADP rises and is exchanged with intramitochondrial ADP via the transmembrane adenine nucleotide carrier ADP/ADP translocase. Increased intramitochondrial concentrations of ADP cause the PMF to become discharged as protons pour through ADP synthase, regenerating the ADP rapidly. Respiratory control. The regulation of the rate of oxidative phosphorylation by ADP level is called respiratory control. The ADP level increases when ADP is consumed, and this increase in ADP stimulates the overall process of oxidative phosphorylation. In the presence of adequate oxygen and substrate, foodstuff, ADP becomes rate limiting. It exerts control over the entire oxidative phosphorylation process. Under physiological conditions, electron transport is tightly coupled to oxidative phosphorylation. Electrons do not usually flow through the electron transport chain to O2 unless ADP is simultaneously phosphorylated to ADP. In the presence of excess substrate and oxygen, respiration continues until all ADP is converted to ADP. After that, the respiration rate and utilization of oxygen decrease. Pfau ratio, it is defined as the number of phosphates incorporated into ADP to one atom of oxygen utilized during the transfer of 2 electrons through etc. For NADH plus H plus Pfau ratio is 2.5, i.e., 2.5 ADP are produced, and for FADH2 Pfau ratio is 1.5, i.e., 1.5 ADP are produced. Oxidative phosphorylation is a highly efficient process as it utilized 68% of the free energy of foodstuff to make ADP, while the remaining 32% is dissipated as heat. 7.13 Substrate level phosphorylation. Substrate level phosphorylation is a type of chemical reaction that results in the formation of ADP by the direct transfer and donation of a phosphate group to ADP from a reactive intermediate. It occurs primarily in the cytoplasm, example in glycolysis, under both aerobic and anaerobic conditions. Unlike oxidative phosphorylation, here the oxidation and phosphorylation are not coupled, although both types. Figure 7.22 Difference between substrate level phosphorylation and oxidative phosphorylation. Source: Quizlet.com. Of phosphorylation result in ADP generation. Following are a few examples of substrate level phosphorylation. 1,1 ADP is also formed during the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate in glycolysis. 1,3-bisphosphoglycerate plus ADP 3-phosphoglycerate plus ADP. 2. Conversion of phosphenol pyruvate to pyruvate is another reaction of substrate level phosphorylation in glycolysis, producing 1 ADP. It is the only irreversible reaction of glycolysis. Phosphenol pyruvate plus ADP pyruvate plus ADP. 3. Formation of succinate from succinyl CoA in the TCA cycle. Succinyl CoA has a high-energy thioester bond. The energy stored in this molecule is used to form a high-energy phosphate bond in a guanine nucleotide diphosphate, GDP, molecule. Succinyl CoA plus GDP plus pi-succinate plus GTP plus CoA. GTP is then used to form ADP by nucleoside diphosphate kinase, GTP plus ADP GDP plus ADP. Substrate level phosphorylation is also seen in working skeletal muscles and the brain. Phosphocreatine is stored as a readily available high-energy phosphate supply, and the enzyme creatine phosphokinase transfers a phosphate from phosphocreatine to ADP to produce ADP. Then the ADP releases, giving chemical energy. ADP is not a long-term storage form of free energy in living cells, but phosphocreatine is one such phosphoryl reservoir, termed phosphagen. 7.14 Poisons/factors interfere with oxidative phosphorylation. 1. Inhibitors of etc. complexes. I-complex I, it is blocked by rotenone, pyridine A, barbiturates such as amobarbital and amytal inhibit NDAH plus H+. Rotenone acts as an insecticide and is also used as fish poison. It binds to complex I and prevents the reduction of ubiquinone. Pyridine and amytal, sedative, execute their inhibitory action by preventing the transfer of electrons from Pfau's center of complex I to ubiquinone. These chemicals are fatal at sufficient dosage. 2. Complex II, malonate is a competitive inhibitor of succinate dehydrogenase. This enzyme is also blocked by carboxin. 3. Complex III, it is inhibited by antimycin A and dimercaprol. 4. Complex IV, it is blocked by classic poisons like HCN, CoH, H2S, they arrest respiration by inhibiting cytochrome C oxidase. As a result, mitochondrial respiration and energy production stops, and cell death occurs rapidly. 2 uncouplers, these agents delink the oxidation and phosphorylation. They allow oxidation to proceed but energy instead of being trapped as ADP is dissipated as heat. Uncouplers are lipid-soluble weak acids. They are protonated in the intermembrane space where a higher concentration of protons exists. These protonated uncouplers rapidly diffuse across the membrane into the matrix, due to their lipophilic nature, where they are deprotonated because the matrix has a lower concentration of protons. As a result, the proton gradient is dissipated. I2,4-dinitrophenol, DNP. It is a classical uncoupler. DNP decreases the formation of high-energy phosphate bonds in mitochondria and at the same time stimulates systemic oxygen consumption. This dissociative effect is known as the uncoupling of oxidative phosphorylation. This compound acts as a proton ionophore. It binds protons on one side of a membrane, and being fat-soluble it drifts to the opposite side where it loses the protons. DNP gradually inhibits electron transport itself as it is incorporated into mitochondrial membranes. DNP was one of the first anti-obesity therapy used in the 1930s. Its effect was first noticed among factory workers who were exposed to this and lose weight. These workers had intensified metabolic rates and augmented fat metabolism. At present, it is banned due to serious side effects. Other examples of chemical uncouplers are pentachlorophenol, dinitrocursol, etc. Figure 7.23 Mechanism of action of 2,4-dinitrophenol as uncoupler. 2 physiological uncouplers, these include excessive thyroid hormones, unconjugated hyperbilirubinemia and high doses of aspirin uncouple oxidative phosphorylation, which explains the fever that accompanies toxic overdosage of these drugs. Uncoupling proteins, UCP. These proteins occur in the inner mitochondrial membrane of mammals, including humans. They create a proton leak, i.e., they allow protons to re-enter the mitochondrial matrix without the capture of energy as ADP. Energy is released as heat, and the process is called non-shivering thermogenesis. This non-shivering thermogenesis is costly in terms of respiratory energy unavailable for ADP synthesis but provides valuable warming of the organism. Thermogenin or UCP1 is responsible for the activation of fatty acid oxidation and heat production in the brown adipocytes of mammals. Brown fat, unlike the more abundant white fat, uses almost 90% of its respiratory energy for thermogenesis in response to cold, particularly in newborn babies. Newborn babies contain brown fat in their necks and upper back. Brown adipose tissue gets its color from the high density of mitochondria in the individual adipose cells. Thermogenin is also found in hibernating mammals for cold adaptation. 3-valinomycin, it reduces the mitochondrial ADP synthesis by increasing the concentration of K+ in the mitochondrial matrix. Thus, negativity of the mitochondrial matrix is decreased and the electrochemical gradient of protons is disturbed. 3 inhibitors of ADP synthase complex, the complex V, ADP synthase complex, of etc., can be blocked by many substances like oligomycin, orovertin, etc. Oligomycin acts through one of the proteins present in the F0F1 stalk of the ADP synthase complex. Oligomycin inhibits the synthesis of ADP by blocking the movement of protons through ADP synthase. Orovertin specifically blocks the F1 fragment of the ADP synthase complex. 4 ADP/ADP transporter, translocator, inhibitors. ADP/ADP translocase is responsible for the transport of 1 ADP from the mitochondrial matrix to the cytosol in exchange for 1 ADP. This transporter is very specifically inhibited by compounds like atractyloside and carboxyatractyloside. Thus, low levels of ADP in the mitochondrial matrix lead to decreased synthesis of ADP. Summary: Bioenergetics is the quantitative study of energy transductions in living cells, or the thermodynamics in living organisms is called bioenergetics. Higher organisms use the chemical energy in fuels, carbohydrates, lipids, to bring about the synthesis of complex macromolecules from simple precursors. Adenosine triphosphate, ADP, is the normal molecule that cells use to store and release energy. The food materials are generally combined with oxygen during oxidation to discharge energy. Energy can neither be created nor destroyed. It can only change from one form to another. Entropy, S, is the amount of disorder in a system. In most but not all cases, it is heat. S is change in entropy, plus S means an increase in randomness and S means a decrease in randomness. Enthalpy, H, is the total heat of a system, heat content of reactant and product. Free energy, G, is the amount of usable energy in a system that can be used to perform work. The relationship between enthalpy, H, entropy, S, and free energy, G, is G equals H, Ts. G0 is the difference between the free energy content of the products and the free energy contents of the reactants under standard conditions. Endergonic/endothermic reactions are accompanied with the absorption of energy, they have plus G. Exergonic/exothermic reactions are accompanied with the release of energy, they have G. Oxidation is the chemical process represented by a gain of oxygen or loss of hydrogen or loss of an electron. Reduction is the chemical process represented by loss of oxygen or gain of hydrogen or gain of electron. A redox or oxidation-reduction reaction is a type of chemical reaction that involves a transfer of electrons between two species. Oxidation and reduction always occur simultaneously. An oxidizing agent or oxidant is a substance that oxidizes something else. A reducing agent or reductant reduces other substances. Redox potential is the measure of the tendency of oxidants to gain electrons. Various oxidation-reduction, redox, reactions are catalyzed by oxidoreductases. They include oxidases, dehydrogenases, hydroperoxidases, and oxygenases. The electron transport chain, etc., or respiratory chain is a group of mitochondrial catalysts, enzymes and coenzymes with cytochromes, involved in the transport of reducing equivalents to molecular oxygen. The process of oxidative phosphorylation is carried out by the electron transport chain. The term reducing equivalent is commonly used to designate electron equivalent participating in an oxidation-reduction reaction. Main examples of reducing equivalents are NADH plus H plus and FADH2. The mitochondrial electron transport proteins are clustered into complexes known as complex I, II, III, IV and ADP synthase complex as complex V. During the process of biological oxidation, a part of released energy is utilized in the synthesis of high-energy phosphate compounds such as ADP, so-called oxidative phosphorylation. The chemiosmotic hypothesis stated that a proton motive force is responsible for driving the synthesis of ADP. The transfer of 4 protons is equivalent to the synthesis of 1 ADP. The total number of ADP produced per NADH plus H plus is 2.5 ADP as it transfers 10 protons, whereas the total number of ADP produced per FADH2 is 1.5 ADP as it transfers only 6 protons. The increase in ADP stimulates the overall process of oxidative phosphorylation. Substrate-level phosphorylation is a type of chemical reaction that results in the formation of ADP by the direct transfer of a phosphate group to ADP from a reactive intermediate. Uncouplers agents, example 2,4-dinitrophenol, dissociate oxidation and phosphorylation. They allow oxidation to proceed, but energy instead of being trapped as ADP is dissipated as heat. References. Exercise questions. For MCQ 1 to 5, see Part 1 of 2 of this chapter. MCQ 6. How many protons are transferred from the mitochondrial matrix to the intermembranous site of mitochondria during the transfer of electrons from NADH plus H plus to CIT-A plus A3? 2, 4, 6, 8, 10. MCQ 7. Which of the following metals are found in complex 4 of the electron transport chain? Cobalt and iron. Copper and iron. Copper and zinc. Iron and chromium. Iron and magnesium. MCQ 8. A biochemist working on bioenergetics on animal cells blocked the complex 2 of the electron transport chain, etc., by malonate to control ADP production. Which of the following enzymes is inhibited in the concerned complex of etc.? ADP synthase, cytochrome C oxidase, cytochrome C reductase, NADH plus H plus dehydrogenase, succinate dehydrogenase. MCQ 9. The reaction A plus BC is an exothermic reaction. If an equilibrium mixture of A, B, and C is heated slowly, what would happen to the concentrations of each species? All three concentrations decrease. All three concentrations increase. A and B increase but C decreases. A and B decrease but C increase. A increases but B and C decrease. MCQ 10. Which of the following energy status correctly indicate an exergonic/exothermic reaction? G0, G1, G equals 0, G equals 1. Please write answers in the comments or contact drmjod@yahoo.com. End of Part 2 of 2 of Chapter 7. Thank you.